Yes,
I
yes,
would like
yes.
to I would like to indicate also to Ron DePauw is the right pronunciation, I think.
Okay, thank you.
Ron can correct me if I'm wrong. DePauw.
Yeah, DePauw.
DePauw.
Pow,
Thank
yeah,
you. Thank you, Ron.
DePauw.
Thank you.
How? Yes. Okay. So we are live now.
Yes, we are live now.
And Shubho, you start recording before you start. Okay.
Okay.
Can I start?
No,
I'll
Shoma
ask
will
you to
Shoma
start.
will ask you to start. Hello, everyone. Welcome to Bioingene webinars. This is our 67th webinar in this series, and we are very glad that today we have a Dr. Ron with a vast experience in his scientific research and different administrative and different collaborative work. So it's our great pleasure to have him in this platform, and I will uh, request all the participants put your questions in YouTube chat box. I will collect it from the Ron and should be relevant to today's talk. If there is any queries about Bioengine, please email us at admin@bioengine.com. After the webinar, we will provide a link for the feedback, and this link will be available in only YouTube chat box. It is actually ensure your live presence or attendance for this webinar. So now I'm request my two friends, Shoma and Priyanka, to help me for this webinar. Shoma will introduce Bioengine, little bit our procedure, and introduce our speaker, Ron. Then we'll go for the talk. Then we'll go for the interactive sessions or interview sessions, which will be conducted by the Dr. Priyanka. And ultimately, we go for the question answer round. So be patient. Don't ask for the feedback link multiple times. We will share it at the end. And those are know about the Bioengine webinar. They definitely know that certificate will available after 3-4 days from the bioengine.com/certificates. And the link is provided in homepage of Bioengine. Our next webinar is for 15. Next one is 19. So registration is ongoing. Shoma, hand it over to you.
Hello and welcome to Bioengine webinars. Bioengine is a non-profit organization created to promote plant science research worldwide. This is an open platform from which plant scientists can present the current advancements of their research to the world. We hope future scientists can gain perspective and inspiration watching the esteemed plant researchers talk about their scientific accomplishments and views on building the future of plant sciences. We are thankful to the speakers and viewers of our Bioengine family for your interest in the webinar series and for your appreciation of our efforts. You can register for Bioengine webinars through our website, bioengine.com, or our social media pages. You are live viewing this webinar and you can watch it again later on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our events. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided in the YouTube chat after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is breeding for sustainable production to meet the challenges of climate change. Our guest speaker today is Dr. Ron DePauw, Science Advisor, SeCan, Canada. Dr. Ron DePauw is an internationally recognized scientist. He has devoted his life to reduce business risk for producers, to meet consumers' requests for safe and nutritious food, to improve end-use suitability factors, and to contribute to an overall betterment of society and the farm economy, including resilience to climate change. Ron was a member of the independent steering committee to the Global Wheat Program of the Consultative Group on International Agricultural Research Management by CIMMYT. He serves on the Borlaug Training Foundation Board, which collaborates with CIMMYT to train the next generation of hunger fighters. Ron has actively participated in the registration of 70 wheat and 6 critical cultivars. For the past 23 years, 40 to 60% of all wheat grown in Canada were derived from the cultivars that he and the Swift Current Research and Development Centre team developed. Cultivars developed by Ron and his team exemplify the use of environmentally friendly and low input cost genetic resistance to control a multitude of diseases and insects. Ron has published 269 peer-reviewed manuscripts, 11 book chapters, and over 1,000 miscellaneous publications, including conference abstracts. Ron has provided leadership to numerous government and non-government committees in Canada and worldwide. He has been invited to review international wheat projects and mentor young international and national wheat researchers. In 2015, he started his own consulting company, Advancing Wheat Technologies, as science advisor to the SeCan Association. His role includes working with breeders, public and private, to bring new genetics to the marketplace. From 1970 to the 1972, he worked on a wheat breeding project sponsored by Canadian International Development Agency in Kenya, administered by the University of Manitoba. Ron worked for Agriculture and Agri-Food Canada for 42 productive years, excelling through leadership and innovation in the wheat industry. He has received numerous awards, including the Order of Canada, the Saskatchewan Order of Merit, Queen Elizabeth II Platinum Jubilee, Gold Medal from the Professional Institute of Canada, which has only been awarded 7 times to someone in agriculture, and an Honorary Doctor of Science from the University of Saskatchewan. We are highly honored that he could be with us today, and we are very excited to hear him speak. So, Dr. Ron, we are all ready now, and we can start the presentation now.
Thank you very much, Soma. I didn't know I had done so many things. I am very fortunate to have very good health and a lot of energy. So a little bit about myself. I have 4 grandchildren, and I'm active with my grandchildren, playing games and also teaching them about growing things. I also have a number of hobbies. I started judo in 1962, which would be before many of our audience were even born. And I'm very active dancing with my dear friend and spouse, Elsa Marie. And we've been dancing since we became empty nesters, which was back in 1992. And we enjoy many other hobbies and hiking, especially going towards waterfalls. We enjoy nature very much. So I was raised on a farm. I was born in Saskatchewan, but raised on a farm in southern Manitoba. And we grew had a large garden, and we grew cereals and oilseed crops, and we had many animals, cows, chickens, pigs, and I would be active. There would be a family activity to be planting a garden in the springtime, planting potatoes there on the left, and I would also be involved in planting and harvesting crops from the time I was a young teenager. And I had the good fortune to go to the University of Manitoba for my Master of Science degree, which I started in 1967. And my chair of my PhD committee was Dean Len Chabeski, that taught plant breeding methods. And as far as I know, it's very hard to find people that are teaching plant breeding methods. And I had the good fortune of being sent to Ciudad Obregón, the CIMMYT winter nursery site in the northwest of Sonora province, in March of 1969, and that's where I met Norm Borlaug and his other people working in wheat research. And then when I finished my my master's degree, Len Shabeski recruited me to join the wheat research team at the Kenya Agricultural Research Organization in Joro. At that time, it was just called the Plant Breeding Station in Joro, and to work on the development of rust-resistant wheat varieties for the Kenyan agricultural system. And Kenya wanted to become independent in wheat. Kenya had gotten its independence in that mid 1960s, and so there was a need for a lot of work to be done to achieve that. And one of the aspects of the work that I did there was to study adult plant resistance on stem rust. And of course, everybody is much aware of what adult plant resistance is. And from Kenya, we had the good opportunity to be growing crops all year round there. But I also, when I returned to Canada, we had a number of locations in Canada, and we grew materials in contra-season nurseries in the Imperial Valley of California, in Obregon, Mexico, Chile, South Africa, and New Zealand. So I've been involved in growing wheat from about 60 degrees north latitude, all the way down to 43 degrees south latitude. And over the course of my life, I figured out I've grown about 100 generations of wheat. So I've seen a lot of changes in the genetics of wheat. So I really feel I'm quite fortunate in these changes. And one of the big changes that has occurred in terms of the development of varieties is that the farming systems for the culturing of wheat has undergone radical changes, from small tractors and tillage equipment like you would have seen when I was growing up as a child on the farm, to these massive pieces of equipment and zero tillage. Single path, where you've got a tractor that pulls a very large implement that will open the soil and place the seed in the soil, and the seed is delivered from this air drill through airflow, as well as there is fertilizer in this particular tank that will then be delivered also, but to separate openers on this particular device. And it's then becomes, what are the genetics that is required to fit this kind of a production system? And you can see that, with the harvesters, a very large header. And so that with that kind of a large header, you want crop that'll stand up and thrash readily. So what kind of genetics was required to fit these kind of production systems? Involved meeting with farmers and trying to sort out what would be the genetics that best fit onto their production systems. So when I started, the typical wheats would be like about chest high. And when I finished, they were much shorter. So the personal goal that I had was to be building new varieties that farmers were happy to adopt. So a semi-dwarf strong straw to fit conservation agriculture became a very big goal. And part of the reasons was that a strong strawed semi-dwarf requires less energy for harvest than these taller ones, because with a tall one, you have to put more biomass through the harvester, which requires energy. And if the crop is lodging, then you've got to cut way down here, whereas in a strong semi-dwarf, you can cut at a much higher height. So it was about speaking about, can we get more kilograms of grain per liter of diesel fuel per hour? And this would increase profitability. There would be less straw to manage after harvest. And also there would be less residue to manage in the spring to prepare a seed bed. So one of the varieties that we released back in 2012, it got into the hands of farmers for the production year of 2015. It is estimated to have saved about 9 million liters of fuel, which would contribute to a reduction in greenhouse gases and air pollution. And the standing stubble contributes to soil moisture by trapping the snow. And I'm going to show you something about this trapping of snow in a moment. So the outline of my presentation will be about climate change, both global and Canada, a background and context to wheat in Canada, and is there genetic variation for heat and drought? Breeding objectives, strategic crossing, population structure and size, and early generation for selection for both quantitative and qualitative traits. And then, of course, you have to have your selection nurseries in hotspots or places in which the genes will express the trait, and you have an opportunity to select for those particular traits controlled by the genes. The National Oceanic and Atmospheric Association has been collecting global land and ocean temperatures for a number of years. And this is the graph that goes from 1850 to 2023. The horizontal line is the average of that 1901 to 2023 time period. And then the annual global temperature is either a positive deviation or a negative deviation. And of course, this is where we are at this time and are very much concerned what can happen, what is going on over here, and how do the plants respond to this? How do people respond to it? How is our world responding to these increasing temperatures? So I was part of a group led by Zhang. We analyzed USDA spring and winter wheat new genetics for 1960 to 2018. That's 59 years. There were 62 sites per year for winter wheat and 30 sites per year for spring wheat, and covering areas of the Great Plains of North America. And then, using climate projections, we used 6 different climate models and 4 socioeconomic pathways. And the results were rather disturbing, very disturbing. It showed that the yields declined by 3.6% per 1 degree of warming for advanced winter wheat breeding lines, and advanced spring wheat breeding lines showed a 7.5% yield reduction per 1 degree of warming. And this is suggesting that future climate warming could outpace the yield gains from our current breeding processes in North America. So drilling down a little bit further, this is now Canada. Wheat in Canada is grown in these dark brown and red areas. It's about 95% of the wheat is grown in Western Canada and 5% in Eastern Canada. And about 95% of the wheat grown is spring wheat and 5% is winter wheat. And that is due to the harshness of our winters, that winter wheat just won't survive. So we looked at what are what has been the weather records in Swift Current, Saskatoon, and Melfort going in this northerly transit, and then projected what would be the temperature and precipitation for annually by 2050. And so this is the annual projected temperature changes to 2050. Swift Current in the south and Melfort furthest north. And the three baseline scenario, the three scenarios relative to the baseline are shown here. And what is really important is what is the difference from baseline as you go through the year. And the growing season is from, this early May to August, so it would be this kind of a time period. And you can see that the temperature will be 2 to 6 degrees warmer in 2050 than currently. And that is about the same as you go north as well. So the projection is that our growing season, which is this period here, our July period is going to be hotter. And that is not good for wheat it is sensitive to higher temperatures. Our winters are going to be milder, and that might make it possible to be growing more winter wheat. When it comes to the projected changes for rainfall, again, the big thing is, what are the deviations from baseline over the course of the year? And for July and August, they will be drier than currently. And so with our growing season, like is from May to August or April to August. And so there's and the most sensitive time period is when the wheat is going through the reproductive phase. So the reproductive phase and early flowering. So there are big challenges for our projected spring wheat. So what are the conditions of growing wheat in Canada? Well, we are a semi-arid continental climate with a high latitude. We have long, cold winters. As a matter of fact, we had snow yesterday, and the snow is projected again today. It's a short growing season. And the growing season is the period between the last killing frost in the spring and the first killing frost in the autumn. And in the case of wheat in our area, a killing frost is minus 2.2 degrees Celsius. Our average precipitation is less than 450 millimeters, with 2/3 of it received in the growing season. So it's dryland production. Conservation agriculture, which is a reduced and zero tillage, has been very widely practiced since the 1990s. And wheat is grown in rotation with other crops like oilseeds and pulses. So dryland production, you need to figure out how to conserve moisture in whatever form it comes. So on the prairies in Western Canada, we look upon snow as a water resource. Just like in other parts of the world, snow in the mountains is a water resource for growing crops downstream. So in the upper left, this is a field that has no crop residue on it. And so there's nothing, when the wind blows, there's nothing to hold the snow back. It blows off into the ditches. Whereas this field on the right, which just happened to be immediately across the road from the field on the left, you can see that there is snow caught in that field. So the stubble that was left standing might have been 20 to 30 centimeters tall, might even have been taller, but it holds the snow in place. And it's quite a uniform distribution of snow. Another technique that farmers have used is developed headers on their combine that will have a raised cutting head that will just take the heads off of a narrow strip of maybe 8 or 10 centimeters wide, and the rest of the crop is cut at a normal height. And you can see with this taller standing stubble, you get like a little snowdrift that here is here. And of course, this will be less uniform moisture across the range. It'll be more moisture here and less over here. So this is the preferred type of snow trapping that is done. And so the farmers want crop residue, a variety that will be strong strawed and stand up erect so that they can leave as much stubble as possible for snow trapping. And then when they have to prepare a seed bed the next year, they don't have excess crop residue to be dealing with. And this shows what happened the next growing season. This is the field that had the snow blowing off, and you can see that the emergence was very erratic, very poor. These plants were placed in dry soil and didn't emerge until 2 or 3 weeks later when a rain came. And so you've got a lower yield, uneven maturity. Whereas this particular field, which was grown in the snow-trapped residue, it's a uniform emergence, and it's going to be considerably higher yielding than the field on the left. So this is just an example of agronomy to contribute to addressing climate change. What are some of the examples of genetic variation to deal with heat and drought? And if we don't have genetic variation, it's very difficult to make progress. So on the left, these 2 spikes of durum wheat are showing a sensitive response to a high heat shock during the floral development, whereas the one in the middle is not damaged. And one of the things that happens is that when the florets are damaged, it is an irreversible damage. You cannot fix it in this growing season. No matter how good the growing conditions are for the rest of the season, the yield potential is punched back to the level of what is remaining in these florets. So finding genotypes that are not as sensitive or more tolerant of a pre-anthesis heat shock is very important. And genetic variation does exist, and people use these kinds of nurseries to find genotypes that identify the ones that are sensitive compared to others that are not as sensitive. Another example is varieties of wheat that have been grown in regional trials in Saskatchewan at about 15 locations per year. So this is just an example of 3 varieties. Brandon, which is a very widely grown, very robust variety. Viewfield, which has also got very good heat and drought tolerance, but it seems to maybe even have a little bit more plasticity, whereas another variety here called Rednet is very sensitive to heat and drought. So in this particular year of 2017, it was an above average moisture year, and you can see that Rednet did very well. But in the years 2019, 2021, when it was dry, Rednet just imploded. And you can see that Viewfield in 2019 showed some plasticity to even outperform Brandon. And this was in 4 of the 5 years. And this is based on 15 locations per year. So there is genetic variation available. So when it comes now to, let's say, okay, we've got a need and we've got genetic variation. So now we should try to be putting that together into a variety, but we want that variety to be grown by producers. So what is the traits that producers want? And I think of that as an ideotype. It's an ideal type. It's an objective. It's something that we want. So we're trying to build something that farmers want. So when it comes to value for cultivation, there's the agronomic performance of yield, maturity, height, lodging, test weight, 1,000 kernel weight. But really important is efficient use of water and nutrients and market grade protection of the harvested crop so that there's minimal spoilage. Resistance to the abiotic stresses. Farmers have to deal with seedbed stress. They don't want something that's wimpy and won't emerge. There'll be heat and drought, low temperature, and pre-harvest sprouting resistance. And of course, there is the grade protection elements and yield protection elements of the biotic diseases. Insects include the wheat stem sawfly and the orange wheat blossom midge, the rusts, fusarium species, bunts and smuts, and leaf spotting pathogens and bacteria. And all of these diseases are important because you can have massive losses if there is an epidemic. And fusarium not only causes loss of grain yield, but also reduces quality through the production of mycotoxins. And of course, when the farmer has produced a crop, he wants to sell it into the market classes that command the highest value, the highest prices. And that is the Canada Western Red Spring market class. It is one of the most expensive wheats grown in the world. It's comparable in price to the Australian prime hards, the U.S. dark northern springs, and hard red winters from the U.S. So there's milling properties, enzymes, starch properties, gluten properties, and baking properties all have to be in there at an extreme level to meet the criteria of the Canada Western Red Spring class. So you might say farmers want it all. They want all of the traits stacked into a variety. And if a variety is missing some of these traits, the farmers will just bypass it entirely. They want it all. And that is really the challenge as a breeder. How do you stack all of these traits into a variety? And so I'm going to shift gears here a little bit to speak about a rationale for making strategic crosses and practicing early generation simultaneous selection for quantitative and qualitative traits. And I'm saying that there's very, very few examples of university plant breeding courses that teach simultaneous selection for qualitative and quantitative traits in early generations. Rather, the norm is you select for the easy to select for traits that are simply inherited, and then get inbred lines, and then select for quantitative traits. And I'm saying, no, do it in the early generations. So I'll go on then to speak about, okay, the traits required to make a top performer that the farmer wants. What are the number of genes controlling those traits? Can you identify the genes controlling those traits? How do you construct a strategic population? And we all are aware that genetic gain is based on transgressive segregation. That is a progeny with the best genes from both parents. It exceeds both parents. And if you don't have genetics transgressive segregation for traits, well, your variety might not be as well adopted as you might like it to be. So here is an example of gene differences between two parents. So this is an example of a cross between an elite and a non-elite parent. The non-elite parent might only have very good drought, or might have a combination of very good drought and heat, but that's all it has. So as a result, you'll have a lot of gene differences for yield. The heat and drought, the maturity, straw strength, the rusts, the various diseases, the quality. So you end up with possibly 77 genes differences between your elite and non-elite. The mid-parent value is going to be considerably reduced from the elite. So to get a transgressive segregation and stack all of the traits that a farmer wants, this is a very, very, very challenging cross to be using. Another example is to use an elite by elite, of which there is complementary characteristics. So what happens then is that there are fewer genes for these various traits that are segregating, and they might have exactly the same genes for certain traits. But you would still get the possibility of a lot of genes to be segregating. But your mid-parent value in this kind of a cross is much higher than in a cross like this. And so your probability of success is better in this kind of a cross, but if there's 21 genes segregating, what kind of a population would you be needing? Another way to explain that or look at that is you have a cross of cultivar 1, which is 2% lower yielding than cultivar 2, which is this high performing commercial cultivar. This one has a little bit of heat and drought tolerance, whereas this one is drought sensitive. And then you top cross it with a heat and drought tolerant genotype, a wonderful new germplasm line. But it's unadapted, it has unsuitable quality profile, poor agronomics, poor biotic stress resistance. And you cross that as a top cross parent. Well, you can see in this kind of a cross configuration, half of the genes are really quite far away from what you want because it's only going to be contributing to heat and drought tolerance. So this would be a cross configuration that does not maximize the favorable alleles. Whereas if we restructure it, we've got cultivar 1 here, which is a little bit lower yielding than cultivar 2, but it's got some heat and drought tolerance, and put our germplasm line with the heat and drought tolerance as an F1 parent, and then top cross with our high-performing commercial cultivar. You can see now that the poorest genes are at 1/4 of the gene frequency, and the better genes are at 3/4 of the gene frequency. But to pull for that, the trait that you want of the heat and drought tolerance, you'll have to be sure to have the right kind of environments to select for that, and not to be pulling along useless genetic material. So I'm going to allude to something here and as desirable alleles, desirable allele combinations. And I'm going to use the simple case of where we have a biparental cross, and the cap A is the desirable allele. And of course, we get the well-known segregation pattern of a 1-2 to 1. But the desirables are these. They're either in a homozygous state or a heterozygous state. It's only going to occur at 1 out of 4. But the 3 out of 4 are actually desirable. And that's what we have to think about when we scale up and why we should be looking at selecting in the early generations. So in an F2 with one gene difference, as I said before, 3/4 of them will be desirable. But as you inbreed, the frequency of the desirables reduces to you have a homostasis at a half each. This is something really important to be thinking about. That the frequency of your desirable gene quotient decreases as you inbreed. And of course, as you increase the number of genes, it's going to increase as a power function. As a power function. As a power function, it decreases. So you can see that, when we said there was a 21 gene difference possible, well, in the F3, it is 0.003. Well, you don't have a smoke of a chance to find it if you wait till inbreeding. That brings up about the frequency of these types per 100. If you have a grow population of 100, well, okay, if there's only one gene difference, you're likely to get it. Or 2 genes, you're likely to get it per 100. But you can see by time you get to 14 gene differences, there's only a couple in 100. And to the 21 genes that we spoke about, the difference between the elite by elite cross, there's nothing there. So this is a big issue. People are growing too small populations. Too small populations. So if you then say, okay, what is the frequency of the desirable genotype per 1,000? Okay, when you're only dealing with a few gene differences, they're there. If you've got 14 gene differences, they're not there in inbred lines. With a 21 gene difference that we had, only 2 are there. And then you've got to think about what is the probability of them actually being present, and what is the probability of your failure to even find them. For that reason, in our breeding program at Swift Current, we would grow 10,000 to 15,000 per strategic cross. And that would give us a chance to have, 20 or 30 or more in the F2, but you have to find them. And the same thing, just another way to think about it for desirable genes, if you are practicing molecular marker assisted. And if you're using microsatellites that are co-dominant, well, in the first generation, you'll get 75. If you wait till their DHs are random in, you get 50 per 100. But you can see how this is decreasing as a power function. So again, it's how important it is to be using larger populations. And this is for the case of 1,000. Okay, that is the background to about the genetic theory about strategic crosses and having large populations. The third element is the environment for selecting. So this is selecting environments from Western Canada at Lethbridge, going across the prairies to Winnipeg in the east. And that is about 1,200 kilometers in an east-west direction from Lethbridge to Winnipeg, all running at that 49/50th parallel. Our annual temperatures are rather modest, and April, May is the growing season temperature. And this is the precipitation, total annual precipitation. And this would be the growing season precipitation, which is maybe 2/3 of the total. And that is fortunate, but the projections are that there'll be less rain in that July, August period. And this is the important one that I wanted to show you over here, is the precipitation deficit. This is the moisture deficit during the growing season. And this is important to have this in order to select for the heat and drought tolerance. But because we are in a dryland farming situation, and these are averages, you don't know what is going to be the growing season conditions. So having multi-locations, you have a much better opportunity or chance that one of these locations will be very stressful and others might be less stressful. But hopefully you've got a good chance of one or more of these being very stressful. And in that way, you'll be able to set up, we've been able to set up a grid of how do the varieties perform as you go under different stress conditions. How do they perform relative to the environmental mean? And then we were able to select always for those that were above the environmental mean and showed the greatest plasticity. We also selected for protein concentration, and we would then regress protein on yield and select for the positive residuals. So that gives us now, we've got the genetics, we've got the populations, and we've got the environments. So then to do a schematic of the breeding program, to run through this, the crosses would be biparental or strategic three-way crosses, or a top cross F1 or a back cross F1. A large F1, and the F2 would be 10,000 to 15,000 plants per cross. And so if you had 50 crosses, you're looking at about 3/4 of a million, 500,000 to 3/4 of a million F2 plants that were space planted. You could see them individually, and we could they were select for bunt, leaf rust, stem rust, yellow rust, straw strength, height, and yeah, but those straw strength, height, maturity have a much lower heritability than, say, stem rust or leaf rust. And the F2, we would select individual F2 plants and grow them as progeny rows in a contra-season nursery, like New Zealand. And yes, we would inbreed, but New Zealand gave us the opportunity, being located at about 42 degrees south latitude, you could select for traits that had a high genetic correlation back with their performance in Canada, like height, maturity, straw strength, shattering, and for some of the APR genes like LR34, LR46, LR67, those genes are effective against multiple pathogens and are race non-specific. So this would also provide us with an opportunity for increasing seed. So the individual plant row would be harvested, and we'd get 250 to 300 grams of seed, depending on the conditions in New Zealand. And that amount of seed would give us enough to grow yield trials in F4 to test for the agronomics at multi-locations, as well as then have separate nurseries for diseases. Prior to harvest, we would collect heads from all of the F4 lines for being able to establish F5 families. So the F4, we would have agronomics and disease, regress yield on protein, and based on that, we'd have about 20% of the population go to the contra-season nursery as F5 head rows as families. The remnant seed from the F4 would be used for predictive quality, milling and gluten strength properties. And we would then discriminate among those F4s F4s on quality. So we would go to the winter nursery then and eliminate entire families that did not meet the predictive quality standard. As well, we would select within the remaining F5 families for the traits that had a high genetic correlation back in Canada, like the maturity, height, shattering, disease resistance. And that would then give a seed to do a repeat then in the F6. And then we would repeat that to the F7 and had F8. And the F8 would give us inbred lines that we would then move to the pre-registration trialing and the registration trialing. And in my next seminar, I'll speak about this going through the registration trialing as we initiate pre-breeder seed and have the breeder seed available when we get to the end point here of a variety supported for registration. If we were doing doubled haploids, the doubled haploid would be produced in the greenhouse, and then we would take it out into an unconfined disease nursery, as we'd only have a few seeds. It might be, 5 to 10 seeds or 20 seeds, depending on what you get from the DH1. And those DHs that showed resistance to the diseases and other morphological traits, we would send them to multiply in a contra-season nursery. And then the DH4 would be the full suite of agronomic disease and quality testing. And then from there, move it into the pre-registration trials and registration trials. So in the next session, I'll speak about how we get this scaled up into the hands of seed growers and into the hands of the producers. So with conventional, this accelerated generation advancement, we'd release a variety in 9 to 10 years. If we use DH, we could shave off a year or two. So when it comes to genetic gain for an individual trait, it's governed by this well-known equation. So this is the genetic gain over time, the selection intensity, selection accuracy, and genetic variance, and the years per plant breeding cycle. So it really is extremely important to be reflecting upon this. Each of these components is very important. Like the selection intensity, well, you've got to have a big population in order to be able to practice a rigorous selection intensity. If you have a small population and you save most of it, well, this selection intensity isn't very good. Your selection and accuracy is also extremely important. The reliability of your nursery, the minimization of errors, the quality of the data collection, the measurement of the traits. And of course, you must have genetic variation. And because you are stacking all of these traits that a farmer wants, you have to have genetic variation for all of the traits. If in your strategic cross, you had everything except adequate straw strength, if it was a weak-strawed variety, no matter how good its heat intolerance was, the farmer will not use it. So, and of course, we want to accelerate generation advancement. And that's where we've been doing this thing about, growing at least 2 generations a year. And using DH technologies. So phenomics becomes very important. This is about nurseries. Whether they're yield trials, disease nurseries, insect nurseries, it's nurseries. So it requires hands in the pans and boots in the fields. Yes, there's things that we can do with robotics. When you think about the things that, the data collection, the way we're doing things, yes. But you will not be able to do this without people committed to doing good work. Genomics, the high throughput genotyping, genomic selection, marker-assisted selection. And again, in here, you'll see there's a lot of stuff about robotics. If you think of the way in which the the DNA analysis is done now compared to 10 or 15 years ago. So it is about people and technology at work. So this particular planter is designed for dryland planting. It has special double disc openers that will cut into very heavy trash, as well as seed bed that might be a little bit hard. It weighs is extremely heavy, and there's packing in front of each of the disc openers, as well as packing wheels behind it. So this dryland seeder has been a major help to be able to get a uniform planting establishment of the plots. And so this would be an example of a breeding nursery. These 4 plots are of the spring wheat, but you'll notice that there is a little gap in between. Oh, my battery is running low. I'm going to plug in my PC. Sorry.
Ron, yes, everything is going smoothly, and
Yes, yes.
in YouTube there is 300 people live watching your webinar.
Okay. Okay, good, good. Okay, so to go back to where I was interrupted with a notification of my battery was running low, you'll see that there is something green growing here in between the plots. Well, that is spring-seeded winter triticale. So it remains vegetative. And that is done by the specialized plant seeder. The outside 2 rows of the seeder have a continuous run of winter triticale that remains vegetative during the growing season. And then this is the target genotypes in the plot. And by having this spring-seeded winter triticale, it sucks up the nutrients, the water, and nitrogen. So it gives the opportunity for seeing what is the capability, the genetic capability of this individual plot. Without having, if the plots were growing cheek by jowl, side by side, then you run having interplot competition. And that becomes a confounding factor when you're selecting for yield and heat and drought tolerance. So this nursery design of minimizing interplot competition and sucking up any nutrients that might be possible around the plot is very important. So with a seeder, they can plant about 2,000 plots per day, a number of harvesters, there's a computerization, well-trained staff, most with a post-secondary degree. So that is about the way in which we ran our breeding program. And we were the only one in Western Canada, the only one in Canada that was doing that. And so now I want to speak about market share. There's lots of varieties that get developed. There's lots of players, and there's about 80 varieties that are currently eligible for grades of the Canada Western Red Spring. So there's lots to choose from. And I started, and this is to show market share over the course of years. So in 1978, I started, and then 1992, 2002, 2012, 2022. So you can see that when I started in 1978, the Swift Current Research Center had about 10% market share. And following this particular breeding methodology, it grew the market share. And some of these others had a lot of difficulty getting going. And this other one decreased. And so others are starting to look at, well, how is it that we have been so successful in stacking all of these traits and producing varieties that farmers want? So in summary, the goal is to stack the traits desired by producers. If you want to have an impact on farmer fields, you have to stack the traits desired by producers, which means that you have to be think about your breeding pipeline to make strategic crosses, but grow large population size. It's the same pipeline, it's just that you've got fewer strategic crosses with larger populations per cross. And I provided the rationale for simultaneous selection of both the qualitative and the quantitative traits in early generations, and the importance of using multi-environments to identify transgressive segregants with plasticity to variable weather, whether there are shocks for heat or drought, or the whole season is hot and dry. But meeting farmer demands results in increased market share. With that, I thank you all for your attention, and I'm most willing and looking forward to participating in discussion. Thank you.
Thanks, Ron. It's a very informative and packed of informations in your all slides, and people are very much love to your informations, because most of the current informations, and I'm sure is very few sources are there where we can get such a huge data. So I thanks to you to present such informative webinar. And for the participants, there is a lot of generics are there. So next webinar also from the Ron in 15th. So if you have any questions in this topic, go through, do some studies, and we'll come again with the questions. Though I have collected few of questions. So I request Priyanka to start the interactive session. And in the meantime, I'll collect 2-3 questions more from the YouTube. So participants, please put your questions on YouTube chat box. Now,
Okay. Shuvo,
Priyanka.
should
Yes,
I stop sharing now?
yes, you may. You must have some. Thanks. Okay.
Thank you so much, Ron, for a very insightful presentation and sharing your wheat journey in Canada. I am sure that many of us will be inspired by your work, and this interactive session will give more insights in terms of your life, your achievements, your challenges, and the involvement you have with various global wheat program, and also recently with the training you are providing to next generation. So to begin with, the very famous and basic questions we ask in Bioingene platform. Can you share with us your journey from growing up on a farm in rural Saskatchewan to becoming an internationally recognized scientist in the field of wheat research?
Yeah, that's a very nice question. As a young person, I like plants and animals. And my father was a hobby breeder. I didn't know it at the time, but he really liked cattle and horses. And, he could speak about, this bull could do such and such, and this cow could do such and such, So I learned from him that progeny can resemble their parents. I didn't know about genes at the time, of course. I had the good fortune to study plant breeding and genetics at the University of Manitoba under Dean Chebesky, who specialized in breeding methods. And he was really a wonderful mentor. And in 1969, he sent me to Ciudad Obregón to harvest genetic materials for my master's thesis, and I met Dr. Borlaug there, and other scientists that I had knew their names from the literature, you know, as a master's student. But the important thing about that was the enthusiasm of the scientists that we would meet. So later in 1969, Dean Schabeski recruited me as a researcher at the plant breeding station in Joro, Kenya. It's now called the Kenya Agricultural and Livestock Research organization. So I went there in 1970, in January of 1970, as a young scientist, and there would have been 6 or 8 other mature Canadian scientists, and there would have been 7 or 8 young Kenyan scientists as well. So here I am. I'm a young scientist with a master's, and there I am with these mature Canadian scientists. So all of the discussion that we would have going on, you know, and we'd be there in the field with the Kenyan scientists and the Canadian scientists, and yeah, so it was really exciting and wonderful time to learn. So that was the beginning.
Sorry, I was muted. So indeed, a very interesting beginning. You got the exposure directly with the mature breeders who can teach the young breeder how to go in the breeding program. So going to the next question, what inspired you to dedicate your career to reducing business risk for producers, ensuring safe and nutritious food, and improving end use suitability factor in agriculture?
Again, I think my upbringing had a lot to do with it. So as a child, growing up on a farm, all the children were expected to participate in the chores, each according to their ability. So as a child, the feeding of the animals, feeding the chickens, weeding in the garden, planting. So I became very much aware of the complexities and the continuous and timely tasks that have to be done. And you're subject to the weather. I can remember in 1953, there was a massive hailstorm that just destroyed our crops and damaged buildings and trees. And my father cried for days and days. It was just a total loss. There was no crop insurance. It was a total loss. So I think this sensitized me to the high risk of farming. Consequently, when I became aware of genetic enhancement and the role that genes play in expression of traits, I wanted to develop superior gene combinations that would reduce business risk for producers. So, like, when the thing about the rust epidemics that kill the crop, this was something I could relate to. I could relate because I had seen a hailstorm destroy a crop. So I think that had a lot of impact on me as to wanting to use science, use the wonders of genetics to incorporate traits that would be able to reduce the business risk. And of course, so if you grow a crop, but nobody wants to buy it, it's a waste. So you have to put that grain, that harvested product, into something that will command a price, and the price will be from the consumers. So you want to make it attractive to the consumers. So it has to be wholesome and nutritious for the consumers as well, so that they will want to buy that product that the producer made.
That's really interesting. So now leading to the next questions, from your childhood to your challenges to see in the family, as a, like how your career went through the reducing business risk. My next questions leads to, as a member of the independent steering committee to the Global Wheat Program, could you tell us about your experiences and the significance of this program in global agriculture?
The Independent Steering Committee to the Global Wheat Program was for the Global Wheat Program 1 and 2. And so it went on for that time period. And under the one CGR program now, there is no independent steering committees. So my role with the Global Wheat Program was 2017 to 2021 inclusive. And the Wheat Independent Steering Committee consisted of about 8 non-CGIR voting members. Okay, so it had to be at arm's length from the CGR system. The members could be from national agricultural research, science sector, the private sector, advanced research institutions, farmer organizations. So it's for diversity of backgrounds and gender. And the relevant geographies for wheat, there was 8 of them. So it'd be Europe, North America. I was representative for North America, Australia, North Africa, Sub-Saharan Africa, East Asia, and South Asia. So there was representatives from, yeah. I remember the people from India as well. Yeah. So anyhow, what we reviewed CIMMYT and ICARTA flagship programs for their alignment and delivery on the UN Sustainable Development Goals. And the ISE role was advisory. So it reported via its chair to the lead center board of trustees. So we reported to the CIMMYT board of trustees. And the wheat program, the Global Wheat Program, was our liaison. So that would have been Dr. Hans Braun.
That's very remarkable. So you could see the differences from the all global wheat programs
Yes.
all over the world, and different challenges they are facing according to
Yes.
their geographical locations.
Yes, yes, yes. Yeah, like the fellow from Zambia was speaking about the challenges of production of wheat and the issues of the heat and drought that they are seeing, particularly higher temperatures. And of course, blast came in during that time period. And that was a whole new pest that had come in. Yes. And then, you know, from South America, the representative, she would be speaking about the need for better information about the quality of the crop for the formulation of the goods that would go into the consumer side. Yes. So it was about improving the grist, improving the quality of the grain that the farmers would be producing. Yeah. So it was very, very interesting.
Very nice. So I would move to next question. That is, you have played a significant role in registration of numerous wheat cultivars. Can you discuss the process of developing and registering these cultivars and their impact on Canadian agriculture? Some of them I already saw on social media, the Brandon and the AC, I mean, these are very famous one, which I see like the spotlight in media.
Okay. I think of the variety registration process as a type of peer review. So as a breeder, I think I've got something really remarkable. It is just top. It should just be able to go directly to the producers. Well, if it is that good, then we should be able to demonstrate that it is. So the Canada Seeds Act is administered by the Variety Registration Office of the Canadian Food Inspection Agency. Seed cannot be sold in Canada unless it is registered by CFIA, that's the Canadian Food Inspection Agency. And the purpose is a form of consumer protection. It's to prevent harm and to assure each variety is unique. So you could say, well, what is the driver of that? Well, the driver is way back in the early 1900s, people were peddling seed without any proof of its performance. So then, of course, when things go wrong, regulations get put in. So that's the background to the Canada Seeds Act. So the variety registration is science-based. So the Variety Registration Office of the Canadian Food Inspection Agency authorizes recommending committees to establish procedures and operating procedures. So in the case of wheat, rye, and triticale, we have a Prairie Recommending Committee that's authorized by the Variety Registration Office to set up operating procedures to be able to measure what is its value for cultivation, which would be its agronomic performance is measured in replicated trials, and its response to diseases is measured, done by the pathologist in their disease nurseries. So if an experimental line shows improvement relative to the checks over the course of 3 years, it can then get a recommendation to be registered by the Variety Registration Office. Simultaneously to this measurement of the agronomic performance, the grain samples from those yield trials are composited and analyzed for milling and baking, the functionality properties by the Canadian Grain Commission. And the Canadian Grain Commission is responsible for determining the market grade. So there's a cadre of cereal chemists and people from the grain sector that will look at the information about the functionality of the grain and make advice then to the Canadian Grain Commission as to its market class eligibility. So when we, at the end of 3 years, we get two significant pieces of information. Its value for cultivation, we know its disease response, and we know what market class it's eligible for. Then, with that recommendation, that knowledge, we can then take it out, make it available, a request for proposals from seed companies to then how would they multiply, promote, and distribute that variety. And I'll provide a lot more detail about that in the May 15th webinar. Okay, so that's how it's done. So and there's lots of varieties that get support to register. Like I think there was this past year there was about 12 spring wheat varieties got support to register, and as I said, in the case of the Canada Western Red Spring class, there's about 80 varieties that are supported for registration. Getting registered is easy. What's complicated is to have the traits stacked that the producers want. Registration is easy. Getting it into getting to what the farmers want, that is like going to the Olympics.
So in that case, if we count total years, how many years it takes for registration? Let's say 3-4 years, and the other to reaching to farmers another 3-4 years. So in total, how many years it takes?
Well, using the method that we did in Swift Current, it would take about 9 years for conventional inbreeding, and if it was DH, we could do it in about 8. And then because we were making breeder seed as we were doing registration, when we got the variety registered, we had to have basic seed. And then from that basic seed of, let's say, 400 kilograms of seed, the seed growers would multiply it up and they might make 20,000 tons if they thought it would be a good one, or they might make more, they might make less. But after 3 years of multiplication with the farmers, then it would be in farmers' hands. But as I said, variety registration is one thing.
Reaching to farmers.
Of the ones that are supported for registration, seed growers might say, we don't want any of those. Because the seed growers, if they're going to start multiplying the seed, they want to know that farmers will buy it. So seed growers are taking a risk, taking this new variety. Will it in fact be one that will perform? So yeah. And in my May 15th presentation, I'll speak more about that.
We will listen more about it in May 15th, that is for sure. So moving to next question about your role in a consulting company. Advancing wheat technology focuses on bringing new genetics to the marketplace. What are some of the key challenges and opportunities you encounter in this role?
Well, my role with SeCan is to be a science advisor and assist others to effectively bring new genetics to the marketplace. And SeCan is a consortium of over 600 independent Canadian seed businesses engaged in seed production, processing, and marketing and retailing. So collectively, the SeCan members are major supporters of seed variety research development and have returned over $150 million in royalties and research funding since its inception in 1976. So what SeCan has wanted me to do is to bridge a gap. So academic training has limitations for everyone trying to become a breeder. A requirement of an advanced degree is to generate new knowledge or technologies. Okay, so it's got to be something new. So my role then has been to mentor young scientists in all aspects of genetic enhancement, from the practical to the theoretical. And that the intention then is to bridge that gap between the practical and the theoretical. And hopefully reduce the time for them to become an effective breeder. And I've got some examples of that, like I don't go and knock on anybody's door, but I am known to be available. And so I'll be invited by young scientists about coming to present seminars. And so we'll work together on, okay, what are your questions? And then we'll work on trying to figure out to answer their questions through seminars. And then I've been invited back and say, okay, what are we going to talk about this time? The first order of business would, what have you implemented of the things we spoke about? And so, yeah, it's been good fun. And yeah, there's some young scientists that have really adopted this breeding methodology that I spoke to you about. Yeah. Anyhow, that has been it's been I think of it as one of the fun aspects of my career is to have done a lot of different things and then to share. No obligation. Nobody. I have this thing about there's many ways of learning. Many ways of learning. And Priyanka, with our children, there's many ways of learning. We'll tell you once, you don't want to do it, learn from your mistakes. You don't want to put mitts. It's cold outside. Put on mitts. No, I don't need to put on mitts, they would say. Okay, go outside in the cold without mitts on your hands. And then they'll say, oh, I'm cold, I'm cold, I'm cold. Give me your gloves. And say, no, you made a choice. So there's many, many ways of learning. Anyhow,
So
that's
that,
off the topic.
yeah, that is also wonderful experience to knowing both sides and connect them and know I definitely to communicate with each other language sometimes might be challenging, but at the same time it's very learning for yourself as you explain with the children example. So going to the next one. Throughout your career, you have published extensively and provided leadership to various government and non-government committees. What drives you to contribute both to scientific research and policy development in agriculture?
Yeah, I get asked that quite often. Like people say, Ron, you're retired. Why are you doing what you're doing? And I say, well, science is my hobby. Isn't retirement about doing what you like to do? So I think, in a nutshell, it's got to do about I have a desire to make a better country. And I believe there should be a strong integration between the pursuit of knowledge and the use of knowledge to make the world a better place. So I strive that there might be food for all. That's very much where it is. And I can remember, in my younger years as a scientist, there would be some very practical people, breeders that would say, so and so has published all of these papers, and if you lit a fire, you'd get more light out of that burning of those papers. So he wasn't impressed with a published paper if it had no impact. And I think that's pretty much what I think about also in that what is the point of this knowledge or this technology if it isn't going to make the world a better place? Somewhere, it's got to contribute somehow to somebody's life to make it better.
That is true. In that case, policy development definitely play a big role to contribute in both scientific research as well as for the public.
Yeah, exactly. Yeah. And if like people ask me to contribute to such and such, well, I think they're asking me because maybe they see that I have something to contribute. So I'm willing to contribute, willing to join in to make a difference. So yeah.
That's a great purpose. And with this, I would like to move to another question. Can you share any memorable experiences from your time working on the wheat breeding project in Kenya, sponsored by the Canadian International Development Agency?
The first memorable memory, is I met my spouse, Elsa Marie, in Kenya.
So your life got changed.
And we are still together, enjoying life very much. So that was one. Okay. From the wheat side, my knowledge of wheat was from a high latitude, 49th parallel north. So we would grow our wheat in the summertime with these very long days. And, it could come to maturity, within 100 days. So I thought that's normal. Well, the equator runs through Kenya, and so there's not much day length change throughout the season, very minimal. So some of the Canadian wheats that grew out in the field, planted in the screened cages, took 5 months for them to even head out, because they had this, they were photoperiod sensitive. So it was really a shock. And of course, then to see this, and of course, then when you are planning your crossing blocks, you have to know something about photoperiod and vernalization. And that was all kind of new to me. So it was, yeah, that was a big one.
Cultural shock.
Yeah, yeah. A second experience that was fascinating was the opportunity to grow wheat all year round. There was no snow. There was no cold weather. We grew 4 cycles in field nurseries a year, and 3 cycles per year using screen cages. So we would start planting in February at a lower elevation, maybe about 1,600 meters above sea level. And then at our main site in Njoro, which was maybe about 2,100 meters above sea level. And then we would plant at a high elevation, Molo up at around 3,000 meters above sea level. And then we could plant it in Njoro twice a year. We could plant in that May period with April, May period with the long rains, and then again in September, October with the short rains. So it was growing wheat all year round. And of course, we all know now, this is a green bridge. So when and there was potent stem rust races there at that particular time. Boy, they could knock out lots. So the UG99 stem rust family, yes, I believe it completely. It was nothing but a green bridge. One of the pathologists did spore trapping, and he could detect spores all year round.
That is all spring wheat we are talking about.
Yes.
Moving to the next question. With over 4 decades of experience in agriculture and agri-food Canada, what are some of the most significant changes you have witnessed in the wheat industry, and how have you adapted to these changes?
Well, there's been many, 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 and I won't enumerate them all, but I'll just give you an example of some of them. I would say adoption of conservation agriculture, this reduced tillage, zero tillage, is massive change, massive changes. It started in the early 1980s, and it became widespread in the early 2000s. And there was a number of things that were involved to make it work. Like you needed completely different planters, openers, the tractors, the size, the scope, the air drills, and a lot of this was possible when glyphosate came off a patent, so that glyphosate was much cheaper to use as a herbicide. Computerization, the GPS systems, those are all very, very, very big changes. So that's on the agronomic side, the production side. So of course, then there was the genetics to fit the changed rotation and these changed tillage practices where required. I would say another massive change is the people that are operating the farms. And I think of this as the professionalization of agriculture. People that are the average, people that are taking over the farms might be children that have gone out and had a professional career, and they've decided, I want to go back and farm. I got lots of examples of that, but I think of that as the professionalization of agriculture. And I've seen it in Obregon, Mexico. I've met people in Tunisia that are representing professionalization in agriculture in Europe. Yeah, it's all over the place that you see very a different mindset about agriculture. Another major change was the farmer levy to support research, which started in 1995. And a farmer levy is the farmers agreed to a deduction of a few cents per bushel, or let's say a dollar per ton, some small amount, to fund research. So and that started in 1995. So in Western Canada, but there's also checkoffs in other areas of Canada and in the United States, Australia, etc. Europe, yeah. So in Canada, about 50% of the cost of wheat variety development is funded by farmers. And this increased farmer funding enabled us to strategically invest in genetic enhancement. We were able to double the size of our breeding programs and developed and used new breeding technologies, such as marker-assisted selection and double haploidy. Like, going back to that 1990s, that's it's developing those technologies. And the rate of genetic yield gain has doubled as a result of that farmer investment and all of those other things that I mentioned. An economic analysis, the most recent economic analysis of the benefit to cost of the Western Canadian wheat levy is 33 benefit to 1 cost. And that is an amazing return on investment.
That is all good side. I would like to know, what do you see which is not good? Because of technology, definitely we are having a double of genetic gain and so many advancement financially as well. What do you think the bad thing happened over the years, which was good in past, not now?
I'm sorry, repeat that, Priyanka.
I'm wondering, what is the bad thing you think, like over the years happening now? The bad thing. You focus all good thing, which is like very good because of the technology. We have a financial, we have a double genetic gain. We have a financial benefits and so many other benefits because of the technology and the other funding. What do you think? What is your opinion for the in the past 40 years ago, the thing were like going very organic and now it is not? So do you think what is your take for bad thing happening now, at that time it was good?
Okay. What has been a negative effect on genetic enhancement? I would say governance. The governance structure. I'll give an example of that, or give several examples. As a young scientist in the late 1970s, I took my science director to the field to show him what we were doing. And I asked him what he thought I should be doing, looking for his counsel. And he said to me, "We hired you to figure that out and to deliver on it." So then things radically changed, so that the governance of science was centralized and controlled and managed by a few, and then they would tell you what you could do. And it's turned everything upside down. And and that is something we really have to guard against, is what will motivate a scientist and what will discourage. And I think as managers of science, as colleagues of science, we have to be prepared to sort out what is good governance and what is not good governance.
Very intense. So now I would like to move to another question. You have received numerous prestigious awards recognizing your contribution to agriculture. Could you share what these mean to you personally as well as professionally?
Well, as I said early on, my goal was to develop superior wheat varieties that a farmer was willing to use his or her dollars to buy the seed and give it a chance to grow on their farm. So I wanted them to get a reward and grow it again. That would make me happy if they would grow it again. If they could say, "Hey, Ron, that was a good variety." That's what I would be happy about. So I was really about focusing on what is it that farmers want and how can we get it there to them and that it'll work for them. So the accolades came as a consequence. I wasn't striving for accolades. Accolades came as a consequence.
Absolutely. This is a byproduct. If you have big goal and these achievements are just the byproducts of these.
Yeah. But Priyanka, there was it was a very dry year. I can't remember which year it was, maybe 20 or 21, 22. I received a phone call from a farmer, and he left a voice message that he said, "It is fun to grow your varieties." I was amazed at how they performed under the challenging conditions. I am so pleased with how it performed. I mean, that's an incredible reward.
Absolutely. So with this, I would like to move to our last question. As a mentor to young international and national wheat researchers, including me, what advice do you have for aspiring scientists looking to make an impact in agriculture?
I would say get to know your target audience. Get to know your clientele. Listen carefully to them and learn what are their issues. Rather than telling them what their issue is and how they should do it, listen and learn. And then to develop a very clear focus on what is to be achieved. And then try to use all of the tools to deliver on that. So that means being a continuous lifelong learner. Like, what are the new tools? Like, there's all of this stuff going on about genomic selection. Yes, maybe these are things that we can use as a new tool. Yes, so gene editing is a new tool. Maybe we can use it, to tweak something that was off a little bit. Yes, so listen and learn and be focused and a continuous learner.
That's really good. And with this, we are ending our interactive and this interview session. And I would like to request Shubho, do you have questions from the audience? Would you like to read them out?
Yes, there is a few questions, but I thought that it is a connecting another webinar, so we'll keep some question for that webinar also, and and also this webinar will be more interactive for the question answer round from the YouTube and all, because we have uh, more time on that day. But now, Priyanka, can you go for the question? The first two questions actually, it is the same person asks two questions, which is related to understanding this talk. So
Okay.
today we are focusing on that, but I am collecting 10, 12 questions.
Okay.
So let's start from
Okay.
these two.
Okay. So I'm beginning the first question from Suren Tiku. He's saying it is an excellent talk. As I understand it, you recommend F2s to be grown in multiple environments and do direct selection of individual plants having desired trait, and then F3 onwards as a families. Is it like that?
No.
No.
F2 are grown as single plants, space planted, and grown to select for leaf rust, stem rust, stripe rust, common bunt, height, yield, maturity, to get some of that. And then to take that plant, and we would apply molecular markers on the seed of that plant, or not. But that's the first generation of segregation. So that would be the plant. That plant would be used to establish an F3 row in New Zealand. And that F3 row in New Zealand was then used to select for genetic traits that relate back to Canada, like height, maturity, straw strength, and APR genes. That would be, that as you know, are effective against multiple pathogens, as well as being race non-specific. With that seed, we might get 200 to 300 grams of seed. We would then make F4 yield trials, and that would be at multi-locations. As well, we would grow seed in FHB nursery and rust nurseries as well. And then from that F4, prior to harvest, we would collect, 5 to 10 heads and just store them. Harvest all of the plots, and that would give us, and we would have the height, maturity, straw strength, all of that, and we would now have the grain samples. And the disease reaction information we would combine that information and select then for those with agronomics, disease resistance, and then we would measure the grain protein content and test weight on those samples. Then we would regress yield and protein to get, the relationship, and we'd select for the positive residuals. With that information, we would then go back to the 5 heads or 10 heads to establish families in New Zealand. With the remnant seed, we would do quality analysis of those that went to New Zealand. And that quality information would be either you're in or out. If you don't have the predictive quality, doesn't matter what your yield was. Doesn't matter what your rust resistance was. If you don't have predictive quality, you're out. And then we would select among the families in New Zealand, those that had the predictive quality, and then select within the families for the traits that relate back to Canada. So that's how we were doing it. So the key point is you can select for all of those traits as soon as you've got enough seed, which was the F4. Like in my master's degree, I demonstrated that there was, you could select for yield in F3, but there was only seed from a single plant. So you can't really do broad adaptation testing. So we decided that, okay, we'll use the F3 generation for seed multiplication to have sufficient seed, then to do multi-location trialing in F4 for both the qualitative traits and the quantitative traits, being the yield and the quality, maybe some people will consider fusarium a quantitative trait, as there's so many genes involved in its control. Hopefully that clarifies that.
Yeah, so never it would be in F2s. Actually, it's very subjective. What is your product profile according to the you made the selection in each generation? So in terms of disease, definitely it goes to F4 as you explained.
Yeah, the
For
disease.
Yeah.
Yeah, for the leaf rust, stem rust, we were selecting an F2.
But not growing in a multiple environment.
No, because you've only got one seed per plant.
Yeah. So moving to the next question, I see that you showed that conventional breeding versus double haploid, the ultimate difference is just 1 year less through double haploid. Hence, what are advantages of using double haploid?
Yeah, that's a very good observation. People think that double haploidy is some sort of a silver bullet. It is not. First of all, if you make a double haploid population and it's not between two elite lines, you've got a whole bunch of junk. Um, uh, where doubled haploidy is very beneficial is you can generate a mapping population. Double haploidy, the gene frequency with double haploidy, you can see it's at a 1-to-1, 50%. You don't have opportunities to recombine like you do when you're dealing with segregating populations, like with straight crosses. So double haploidy, you have to think about, what am I using it for? Am I using it for a genetic study to make a mapping population? Or am I targeting to make a variety? And if you are, then you have to be darn sure the two parents are both elite and have very complementary genes, that nothing is missing between them. And you might be able to practice marker-assisted selection to advance it. And yes, we have released a number of double haploid varieties out of our breeding program at Swift Current. I think it is about a quarter to a third of the area is planted to double haploid cultivars. But you got to really think about double haploidy as a strategy, how you're going to use it. It's not a silver bullet.
So just these two questions. We have so many questions, but next webinar, we don't have interactive session. So we can take those questions. So that would be good. With this note, I would like to end this session, and I would like to ask Shubho, can you please give concluding remark?
Thanks, Bianca. Thanks, Ron. Is a great, and people are all also they are online and asking many questions more and more, and we have received many appreciations. I can little share you what is as you said that your excitement is the when you got the call from the farmers about your success, and like here we also got uh, many appreciations on your talk and all. But next, what we try to do in the next webinar, we don't know how many people are actually watching, but these 363 people are apply for the certificates and participations, and all are uh, appreciate your webinars. So I will send you the this link, and these are the people. This is the live form. These are the people from different countries like Bangladesh, Benin, Canada, China, Ethiopia, Ghana, India. These are all just watched and submit their forms. Indonesia, Iraq, Nepal, Nigeria, Pakistan, Philippines, South Africa, Spain, Sri Lanka, Southern Indonesia, United Kingdom, India, and maybe it is ongoing. So again, India is started here. So we appreciate your involvement. We appreciate your help to all of us, and these people from our those are the regularly viewing our webinars are very happy for your webinar. And yes, next day we will discuss something else, like we wanted to know about your point of view on collaboration work ethics, manuscript writing preparations, and on time delivery, because time we are I don't know, I am very bad management of time, so maybe on time how to deliver. So we will focus on that discussions, and we will do your webinar then discussion session, not the interview sessions. Today is a interview session. That day we will discuss these things, because we are very excited from your knowledge on the subject and up to date knowledge, like you mentioned that after retirement to keep into that. And that day we also ask you about the speed bidding, because in wheat people are using the speed bidding. So about the speed bidding technologies, because it is very new to us, means it's in India uh, 2-3 locations is installed, but I don't know how it is functioning, and all for the crops, specifically wheat and maize, like that. So that day we will focusing on that aspect. So
Sure. Okay.
for today's is a great, and as I mentioned, all the viewers that this webinar is, the recording is available. Go through the slides, make your questions. So next time we'll go for more time on question answer round, and today we'll pick many questions from the YouTube, which will be answered in next day on 15th. And great talk. Thanks, Ron. There is a 350 people's like lively watching, and I think the viewers is more than thousands right now. We'll update you about all the update we have gathered today's webinar, and we are waiting for your next webinar on 15th.
Well,
So
it was a real pleasure for me to be invited to do this and to prepare for it and to share with people from around the world. This is very much who I am and how I like to be with people around the world. Yeah.
yes, and thanks Bianca for arranging this and request Ron first to there and convince him, because we are very new, so convincing is all. We need to convince people, and that's before starting of the webinar we're talking about the bioengine and all. That is I'm that I was told that we are trying to bring a community plant loving people. Though today I got the 2-3 suggestions to bring the animal science, cancer biology, and nanotechnology like things. But yes, plant bioengine is for the plant science, mainly for the plant science background, and our target is in that way to bring the many renowned scientists in this, and we try to gain the knowledge from them, and it will be recorded so anyone can see it in the future. So thanks, Ron. We are meeting again in 15.
Yeah.
And
Thank
yes,
you, Ron.
one comment is there that your smile is very attractive.
Well, thank you very much. Thank you. And I
That's
look
true
forward
indeed.
and I look forward to preparing and sharing with you all again.
So
Thank you so
yes,
much, Ron.
yes, and these all comments are also available with the this recording, so people can watch and enjoy once again.
Yeah.
We have really enjoyed your webinar. Yes, we really
Okay.
enjoyed.
Yeah.
Thank you.
Bye.
Bye-bye, everyone.
Bye. Thanks, Bianca. See you in 15. Okay.